My name is Ikenna Kingsley Uchendu. I am discussing the topic Role of Protein Kinase C in CD47 mediated phosphatidylserine expression pathway by Bric 126 in leukemia cell line. Leukemia is cancer of blood-forming tissues, including bone marrow. Many types exist, such as acute lymphoblastic leukemia, acute myeloid leukemia, and chronic lymphocytic leukemia. Many patients with slow-growing types of leukemia don't have symptoms. Rapidly growing types of leukemia may cause symptoms that include fatigue, weight loss, frequent infections, and easy bleeding or bruising. Treatment is highly variable. For slow-growing leukemias, treatment may include monitoring. For aggressive leukemias, treatment includes chemotherapy that is sometimes followed by radiation and stem cell transplant. Treatment failure in T-cell acute lymphoblastic leukemia occurs when leukemic blasts acquire resistance to chemotherapeutic agents. Current research efforts are focused on the search for targets for the development of more effective and less toxic anti-leukemic drugs. CD47 has been suggested to be involved in chemoresistance and cell metastasis, although several potential mechanisms were suggested to explain the therapeutic effect of CD47 targeting. The downstream effectors which lead to different effects by CD47 are still not well understood. The aim of the research was to investigate the role of protein kinase C in CD47-mediated phosphatidylserine expression pathway in Jurkat T cell. Method: the cell lines and antibody. Jurkat cells were purchased from ATCC in Meadows, UK. Bric 235, which is a mouse immunoglobulin 2B anti-CD47 monoclonal antibody, and Bristol, which is mouse immunoglobulin 2G2B anti-CD47 monoclonal antibody. They were both purchased from the International Blood Group Reference Laboratory in Bristol, UK. Chemical reagents used were the PKC inhibitor known as the bisindomalamide 1 hydrochloride, ROPMI 1640, Hanks valence salt solution, HBSS, fetal bovine serum, trypan blue stain, propylidium iodide, annexin V50, and binding buffer. Cell preparation and apoptosis induction. Cells were washed 3 times with 1 ml HBSS at pH 7.4 and were spun between each wash 3 minutes at 200 G to remove any ROPMI or metabolites so as to avoid any interfering protein. The counting of cells was completed with a hemocytometer and a standard inverted microscope, and cells diluted in HBSS buffer at a concentration of 5 times 10⁶ cells per ml using a flat bottom 96 well plate. Jurkat cells at a concentration of 5 times 10⁶ cells per ml in 200 microliter of HB were incubated with 5 microliter of 1.3 mg per ml Bric 126, 5 microliter of 1.2 mg per ml Bric 235, or control overnight in separate wells at room temperature with gentle agitation at 36 revolutions per minute on a SWAT gyro rocker to enable even distribution and binding of antibody reagent to the cells. In separate wells, Jurkat at a concentration of 5 times 10⁶ cells per ml in 200 microliter were first incubated at 37 degrees Celsius with 1.5 microliter of BIS1, which is a PKC inhibitor, for 30 minutes to allow the inhibitor to penetrate the cells. Cells were then incubated with 5 microliter of 1.3 mg per ml Bric 126, 5 microliter of 1.2 mg per ml Bric 235, or control overnight at room temperature with gentle agitation at 36 revolutions per minute on a SWAT gyro rocker. Control cells were incubated with HBSS buffer only. Results: the histogram A shows the mean percentage annexin V positive cells. It was observed that cells treated with Bric 126 exposed more PS than cells treated with control and Bric 235. Now, the histogram in B shows the mean percentage number of viable cells in the 3 groups or treatment groups. Now, cells treated with Bric 126 showed less viable cells, that's more dead cells, compared to cells treated with Bric 235 and the control. This is the PS exposure and apoptosis level measurement after PKC inhibition. Histogram A shows the mean percentage annexin V fit. Figure 2 shows phosphatidylserine exposure and apoptosis after PKC inhibition. Now histogram A shows the mean percentage annexin V positive cells. Now in histogram A, it's shown that cells treated with BIS1 had more PS exposure than cells treated without BIS1. And also in histogram B, it was shown that cells treated with BIS1 had lesser number of viable cells, that is, showed more dead cells than cells treated without the PKC inhibitor. This is the flow cytometer profile showing PS exposure in Joker T cells, which supports the histogram that was earlier explained. Discussion. CD47 ligation with anti-CD47 monoclonal antibody caused the exposure of PS, which is consistent with previous studies. Cells treated with Bric 126 showed a higher PS exposure than Bric 235 or control. This demonstrates that CD47 molecules are expressed on the cell membrane of Joker cells, and also supports previous study that ligation of CD with anti-CD47 monoclonal antibody induced PS exposure, hence apoptosis in these cells. Percentage viable cell count was done to find if PS exposure with or without PKC inhibitor correlates with cell death, hence apoptosis. It was observed that cells treated in the presence of BIS1 had lower mean viable cells than cells treated without BIS1, although not statistically significant. This again suggests that PS is a measure of cell death. Results from this experiment demonstrate the involvement of PKC in CD47 PS exposure pathway in Joker T cell. This is worthy of note for future work. Conclusion. Inhibition of PKC pathway promoted anti-CD47 induced apoptosis in Joker T cell. There is need for identifying more effective anti-leukemic targets or drugs. The observation from this study identified CD47 and PKC as novel functional proteins in Joker T cells with promising therapeutic potential. This study would provide insight for targeted therapy against T cell acute lymphoblastic leukemia disease.